Should we share again?
Oh, there you go.
Okay, cool. All right. Hi, everyone. Good to see familiar faces and those who showed up on Zoom. Thanks for coming today. Today, I'm going to be talking about inferring gene regulatory networks from gene expression data. My name is Negin. I'm a fifth-year PhD candidate in Vivek's lab. And these tools are some of the tools that I've been using for my own research and wanted to maybe get some of your feedback if you've used them or if you see something in my approach that I can take in, or if I can maybe show you how to use these tools, that would be great. Okay. So the overall theme of this talk will be in 6 steps. First, I'm going to be talking a little bit of the overview of what do we mean by GRNs, and then what are the existing GRN tools. And then I really enjoyed using pySCENIC because it was very fast and easy and straightforward. But there are some limitations to using pySCENIC. And then I'll talk about how introducing priors can actually help guide the GRN reconstruction in a more biologically relevant way, and how we use this other tool called NetREm to complement the SCENIC results. That'll be step 5. That I'll show how I integrated both of those methods. And then the last step is more about diverging from these classical ways of inferring GRNs and more about the deep learning models that are all hot these days. Okay, so taking it all back to our cells, we know that every cell has the same DNA, and in the nucleus, RNA transcripts are made from the DNA, and then we have the proteins that ultimately guide the transcription of a specific mechanism in the cell type. But even with the same gene, we get different cell types. So we get neurons, we get astrocytes. This is in the brain, of course. We have oligodendrocytes, we have microglia. So what causes these different cell types to emerge? Is it just one gene? Not really. There are a bunch of genes, basically, that give us the expression profiles of a cell type. I'm saying cell type here, but you can think of disease profile, you can think of any other biological mechanism that you'd be interested in. Normally, it happens through concerted activation of multiple genes together. So many genes basically are involved in determining a cell type specific program or other type of programs. But another important thing is these genes do not act independently. They're acting together. So how can we know how do they affect each other, or how are they coordinated together? This takes us to the principles of gene regulatory networks. And why do we care about these? Because, well, a lot of the data that we have is just single cell RNA expression data, which, as we know, is when the mRNA is being made from a gene. However, there are other modalities like the ATAC-seq data, where it helps us know the open chromatin structure that help us kind of build a better network for our kind of gene expression data. So we know about the cis-regulatory elements that are these non-coding regions in the DNA, and they basically help bringing different parts of the genome, especially the promoter region or other regions, together so that the transcription factors can bind. And when they bind, they bring these other proteins there that ultimately brings in the RNA polymerase, and then we have our expression going on. So you can see, like, if we have more information about these even non-coding regions, that can really help us make this network. So classically, we show the GRNs with the nodes and edges. So we have the nodes that are either transcription factors or genes, and then the edges really show the kind of the direction of the interaction. Well, many times we don't even know the direction. We just know that there is an interaction between these proteins or the transcription factors in the genes. Many of the times that we use gene expression data, we have the matrix, and then based on only the expression value, we can find like correlations or associations and kind of determine how those genes are interacting together, maybe like being upregulated or downregulated. Where you see the chromatin accessibility in that diagram is where we normally, well, nowadays we have it more because of the technology, but many of the times we just have the like gene expression data. With the single cell RNA-seq and other modalities, now we are able to infer many of these GRNs that not only are cell type specific, but now we can also in developmental studies, we can understand if a cell type is changing its regulatory program, maybe becoming like from normal state to a disease state. We see it in microglia states that we can kind of see what genes are changing throughout the course of the disease and kind of have an understanding of the cell type behavior. And then on a more, I guess, classical level, we can find the disease programs in patients with a certain condition versus the healthy individual. So basically the GRNs help us understand these programs that go beyond just disease versus normal. We can also understand cell type identity or how basically cell types just change their states in development or other disease status. So there's a lot of use to them. Now, to infer the GRNs, there have been many tools. I'm putting this table there because I'm trying to show that even though there are a lot of tools, you can see the columns, the type of modeling is different. So some are linear, some capture nonlinear interactions. The statistical framework is often not the same. So many models have their own assumptions that may not be relevant to your data set. And also, if you care, like the downstream of the distance from the promoter or where the gene is starting transcribing, all of those are assumptions that all of these models are making. So yes, there are many tools. And you have to be aware that expression data alone can be noisy and is basically just not enough. So we have to have a lot of assumptions, or we have to kind of fortify our gene expression data with other modalities so we can make these networks in a way that is sensible or biologically relevant. And many of the tools do not use priors, at least the ones that I am showing here. So even though they change in the framework they use or in the type of modeling, they really don't incorporate any other modality in their modeling. So just putting SCENIC there because it's very popular and it's a very well-developed suite of tools for not only just single cell gene expression data, but also other modalities like ATAC-seq as well, that our very dear friend Tatiana and MCSB alumni gave a talk on using SCENIC Plus because she had ATAC-seq data, so check her talk out for sure if you're interested in using both a single nuc RNA-seq and ATAC-seq to understand GRNs. But for my data, I am basically having like an atlas of basically many data sets that I have integrated. So I have a lot of gene expression data, RNA-seq, single nuc RNA-seq data. So I wanted to learn about the different programs in my data, and I wanted it to be fast and kind of give me an idea of how my data looks like or the regulatory programs look like. So I opted to use this tool called pySCENIC, which uses a lot of steps from SCENIC. And I'm going to briefly kind of go over the steps here. I tried to make it as concise as possible, but I think it's still useful to understand the steps behind it. So panel A shows in the first step, SCENIC takes the gene expression matrix and builds co-expression modules based on just the gene expression. And it uses either Gini3 or gradient boost, which are two ensemble method modeling ways to predict these TF-TG interactions. So once we have the modules, then the next step is to kind of bring in information about motifs, enrichment that has, like, this data sets have been, or I guess experiments have been done to understand what transcription factors bind to what motifs. And that's where you see in panel B that every gene is binding, or at least every gene has a promoter, and that promoter has a sequence that a transcription factor will bind to it. So these sequences are different for every transcription factor. And in the third step, which is to understand the cell scoring, is basically once we have formed our transcription factor target gene regulon. So a regulon is a transcription factor and all of the genes that it is basically activating or is associated with. Then we can measure how much that regulon is active, or how much the transcription factor is affecting all of those genes. And pySCENIC let us know of the activity using this AU cell method, basically. I'm gonna like show how the result also looks like. So it's like kind of a little vague here. It's gonna look a little bit better later. And then we have the clustering, where you can merge the activity scores with the rest of your data set, and you can look at it on a UMAP or a t-SNE, or however you prefer to look at your data. One thing to run SCENIC is, aside from gene expression matrix, we also need external resources or external data sets. So as you can imagine, we need to have the motifs and the binding sites and the list of transcription factors. So this SysTarget is a repository that has ranked the genome based on their motifs. And pySCENIC also provides a list of transcription factors for both human and mouse, so it's very easily downloadable. Once you have the external data sets, you just run the steps and you get the regulons and activity matrix, as we discussed. Again, so now I'm just showing like what you expect to get at the end of running every step. So step 1 was running the default method is the gradient boost. And after you run that, you get this TF-TG co-expression module, which is mainly a candidate list of transcription factor target genes. The importance score is really just showing how effective that transcription factor is in predicting the expression of that target gene. So it's not to be taken as like a coefficient. It's more like there is an association between these, and higher importance score basically means like there's a higher chance that this transcription factor is affecting that target gene. And then in regulon prediction, we are basically pruning the list of TF-TGs based on which of these transcription factors have a motif that was recognizable in a promoter of a target gene. So if there was no match, we basically drop those TF-TGs, and we end up with list of more limited transcription factors and the genes that it's regulating. So at the last step, we measure it's like an AUC score that shows how those target genes together are activated by transcription factor. And it's really like a score that I guess the higher it is, like you don't have to make sense of it a lot, but you just have to know that the higher that activity score is, it means that those genes are more acting in concert in response to that transcription factor. So this is just one example of showing how FOXP2, which is a transcription factor in excitatory neurons, are being activated. So the regulon is just, the panel on the left just shows the expression profiles. So the expression levels are, which is counts. This is not normalized, just the counts. The expression levels are not really that telling, but if you look at the regulon of that transcription factor, you see that they're kind of acting quite in concert together. So the transcription factor is having an effect, even though we don't infer much just from the gene expression alone. All right, so that's a very simple way of putting the steps needed for pySCENIC to run and kind of the outputs that you can get. Now, why running pySCENIC in alone was not enough for my data set? I'm only using RNA-seq expression data, right? Gene expression is high dimensional, and by extension, transcription factor gene expression is happening in a high dimensional space. And we don't know necessarily if biological data or in that biological realm, also we have that much of dimensionality. Maybe we need to like really constrain our domain when we are looking at these transcription factor and target genes. And we know that the expression data is noisy and it's sparse, especially in single cell data. And also, transcription factors also are affecting each other. We don't really consider that in SCENIC. We just think that there are target genes that are coordinated together, and there's one transcription factor that is affecting them, but transcription factors also are coordinating each other. And we don't really have any directionality when we have like a TFTG or TGTG. Well, that's not really that matters, but we only know there's a link, there's a connection, but we don't know if the transcription factor is activator or repressor in those inferential modes of using SCENIC. So if we use priors, and by prior, we just mean like a data that we know that is validated through either biological experiments or literature-based research that we know is something that should be there. So if we include, let's say, like a network of protein-protein interaction, which means like transcription factor interaction, then the GRNs that we get also make a little bit more sense. So if we include those information, we reduce spurious associations, and we kind of guide the regression or the estimation of making these networks. And if we do see new links, then we have a little bit higher confidence that the new things that we find make sense biologically. Also, if we are interested in a specific cell type regulation or a specific condition, and again, we know that certain transcription factors should be there, this is a good way of limiting or kind of looking more closely to something that we find interesting biologically. So the GRN inference now becomes less about just finding modules, because you can find any modules, like you can find groups of things that work together, but it's more about understanding there are constraints in biology, and if you incorporate more information, you can understand networks that are like, they matter basically. So we collaborated with Sonia, talented postdoc now. She's in Cambridge, and she developed this tool called NetREm, Network Regression Embeddings, that is still using some of the more classical ways of understanding TFTG, but she allows for incorporating these protein networks prior. So I'm going to try to explain her method, but I'm sure she'll do it like way better, but I'll try my best. Basically, the idea is we have this transcription factor expression. So we have our single cell or even bulk expression data. But this time we have transcription factors. We know what our transcription factors, and we have a target gene. So a little bit different here. Now we have one target gene, and we have a series of transcription factors that we think they are coordinating together to regulate that one target gene. So we need the transcription data. Then we need a protein-protein network interaction, and then some prior knowledge. And then there's a two-step calculation process to infer this gene regulatory network. And at the end, we come up with kind of known networks that we can also find some novel findings. That's an overview. To kind of go a little bit more in depth of each kind of step or the input and like the processes. As we said, we need the gene expression data. This is really for her purposes because she's not only just inferring, but she's doing like a predictive modeling. So once we have the single cell or our bulk expression data, she is kind of using her training and testing data set. but the goal is to really, oh, I think I'm blanking. Yeah, so I think the goal of this slide is just to show like you need the gene expression data. You can decide however you want to split your data set if you want to do the predictive modeling. But every row is a sample or is a cell, and every column is a transcription factor expression. And basically, this is to show I thought the cartoon is pretty funny, is showing like, yeah, you can have all sorts of expression happening, but you need to kind of give it some information to understand what it is that you're trying to look for in the data set. Otherwise, you have all of these gene expression data, and you can find all sort of association, which is then why she incorporates this protein-protein interaction network. So this is a network that she has stimulated. We have an example of 5 transcription factors. We know that TF5 and TF4 are highly correlated. This is based on, again, like string database that has validated experiments that are known based on a particular condition. So we know TF4 and TF5 are in direct communication or highly affecting each other. So as a second input, we try to incorporate that also into the model. And then later we show how mathematically she tries to go about that. But for now, you have the input, the gene expression, and you have the xj, the network next to it, which is a transcription factor, and their association and the weight between them. So the weight is really, again, based on the string database or other known interactions. So if I know that TF4, TF5 have a high weight, we try to incorporate that into the model. And then the third input, which is optional, this one allows us to give it like a TFTG, a known, or like a previously inferred transcription factor target gene network. This is where if we combine, which is a framework that we use, if we combine like tools like SCENIC to have like an idea of the transcription factor target gene interactions, then we can actually incorporate this into NetREM's framework. So the final network that we get kind of is more constrained based on all of these known interactions. And if no prior knowledge is used, then we are just basically using all of the transcription factors that are known in that cell type or in that condition or whatnot. And one thing to take note here is that the steps are happening for every single gene. So if you have how many ever genes you have, this NetREM will run through all of the transcription factors for that gene. So you have a network of really like connecting how transcription factors may be like regulating genes, and get a network for that. So just a little bit getting to the math of it, just because this is GenPals. I don't know if some people may be into the methods part. I'm gonna like expand a little bit on it. So now that we have all the input, we have the matrix, we have the PPI network that we use as a prior, and we probably have some TFTG known interactions from before. Now, what we are interested in, we are trying to find ways to model this network in a way that okay, how do I say it? So there's like 3 parts to this. The very first part is we fitting the data to a regression-based model. And we are trying to basically reduce the loss of prediction. So whenever we predict, we have some loss, and we're trying to reduce that loss. So the first term is that regression-based data fitting term. And then the second alpha hyperparameter, what shows is a sparsity prior term. So if you're familiar with the lasso regularization, basically when you have so many variables, in this case we have a lot of transcription factors, and we know that maybe only a few of them are necessary or interesting for our biological question, we try to shrink the other transcription factor weights towards 0. So the higher alpha is, the higher the penalty, and basically the sparser the network will be, meaning fewer transcription factors we're going to consider in the network. Another hyperparameter that we can tune here is beta. And beta is based on the previously known PPI interaction. So the transcription factor network that we have, kind of with beta, we can decide how much of that interaction we want to save. So the higher the beta, the more we are respecting the known transcription factor association. So I guess the ultimate ideal GRN based on this framework is one where you have a few well-connected transcription factors that are able to kind of predict the expression of this target gene somewhat in a consistent way. So we already talked about the overall input on step one. So why NetREm kind of help us in this inference is, if you look at the panel on the right side, we see the base of a set of correlations for target gene Y based on transcription factor 1 through 5. You can see that transcription factor 5, if you take the absolute value, is highly correlated with target gene Y, so the expression of a target gene. And if you remember, we already know that a transcription factor 5 is connected with other transcription factors. If we don't see the network on the bottom and we only look at the correlations based on the gene that are based on just the gene expression values, we can see TF3 is slightly higher in correlation compared to TF4 with target gene Y. So you may think that TF3 is probably more associated with target gene Y. So we can consider that, right? And then if we look a little bit closer, target transcription factor 3 that has a slightly higher correlation with our target gene is like a lone wolf, is just stranded there, and we don't really know exactly how it is functioning in a biological network. So what NetREm does is it's also considering that high affiliation with a known transcription factor that is known to have a high effect on gene expression of Y. And based on that gives a higher weight to transcription factor 4. So if you just were going to go based on the correlation, as you can see, with these other models, you would think that transcription factor 3 is slightly higher in correlation. So it's affecting the target gene Y a little bit more. However, transcription factor 4, even though it has a slightly lower correlation with target gene Y, it's weighted so it has a higher effect in our final predictive modeling. So this shows that having a prior, considering a prior in your GRN inferential can make it a little bit more biologically relevant. And we can, if you want to think about it, is more like if you're in an institute and you have the dean, and you know the dean that works with particular labs a little bit more based on a research subject. So if you know the dean that works in a particular subject, then you know by proxy other research labs that are working on a particular topic. So it's more like knowing something that is validated through biological data, and then you find other things by proxy that you know makes sense. So you don't just carry whatever that shows up as important just by correlation. So at the end, running NetREm gives us two important things. First, we get the TFTG coefficients, and there is a directionality to it. So I know if a transcription factor is activating a target gene or is repressing a target gene. But also, we get a TF-TF coordination network where we can understand how these transcription factors are also affecting each other in making that network. And we also have some novel links here that we can now infer. But this is building on top of what is already known. So TF4, TF5 was already known to be interacting. So if you have like new links here, then you can like trust that there's something biologically relevant there. Again, beta is the hyperparameter that helps us tune or helps us understand about the TF-TF interaction. So if it's higher, if it's positive, then it's cooperative between them. And if it's negative, then it's antagonistic. So they're working in the opposite directions. And the bottom panel is basically just showing the outputs, but more like edge and node representation. Again, we get this a TFTG network on the left side, with C being the weight that determines the direction of interaction between transcription factor and target gene. And on the right side, you get only transcription factor networks. All right. Now, how did we incorporate it in our data set or in our model? Was basically through this workflow. We already had the single cell RNA-seq data. We had to first run pySCENIC to get those TFTG candidates. If you remember, TFTG, pySCENIC does give a TFTG adjacency matrix by which we then determine the regulon, and the next steps was kind of like just specific to pySCENIC. But we have to run pySCENIC just to get the TFTG candidates, which then we will feed into NetREm as one of the inputs, and to make the networks a little bit more consistent or more robust. And not only that, but we still need the matrix, the gene expression matrix for NetREm. So we have those, and then we also have like Jasper or other databases that help us know about the TF motif interactions. Also, we need to provide NetREm. I guess like you can always give more information about the TF motifs, and then also the TFs that are relevant to you, either is a particular disease or humans or whatnot. You can always like supplement NetREm with those. So after getting the results from NetREm and from pySCENIC, we already have lists of TFTGs. So now it was a matter of finding what was consistent between the two of them. So we went with the ranking-based approach of finding the 5,000 top TFTG interactions from both of the methods. And then kind of just cut it down to the top 300 TFTG links and try to focus on that for each cell type. And this way we have a more reliable or robust transcription factor target gene network that also gives us directionality of how each transcription factor is affecting the target gene, but also we know how transcription factors are affecting the other transcription factors. And basically it's a signed network. So just as an example of how this worked, you can see on the y-axis, we have a list of transcription factors in astrocytes in Alzheimer's disease. And the y-axis is all the target genes, not all the target genes, but series of target genes for each of the transcription factors. And what we found that was kind of consistent with what we know that happens in astrocytes in Alzheimer's is this interaction between this JUN and BOS transcription factors that was captured through this framework. And if we do try to visualize the network and all of the interactions, even though it's pretty busy in here, but you can see that in the final network that we get, this JUN and FOS are, they seem to be kind of close together, and they have a lot of positive or cooperative interactions with others. And a little literature search showed that these two actually are forming like a heterodimer, and they are known to be involved in disease-associated astrocyte in Alzheimer's. So I guess this framework helps us detect things that have like a meaning, or at least are showing up in other literature. And based on that, we can kind of explore other possible interactions, especially like there was a bunch that I was just grabbing my attention, but probably that's more biology related. And I wanted to end the talk with saying how now all of these AI tools and these deep learning models are really becoming the new norm. So everything I discussed about the NetREm and pySCENIC, they really rely on these kind of, to me, like more older machine learning methods that are still very plausible, but they are still linear based. So they may not capture all of the higher order interaction between the different kind of data sets. And also these deep learning models allow for involving or integrating all of this like ATAC-seq, ChIP-seq, RNA-seq modalities together. But I think it's still very useful to understand exactly how biology comes into the to the interface, because you can use any of these models, but then again, you can be grabbing onto whatever that the model shows. So being aware of the like incorporating these priors and how you need to have some sort of constraint helps making your results more interpretable. I didn't get a chance to like complete it, but these are just a bunch of like tools that I guess for another talk we could get into them. But yeah, that's the end of my talk. Thank you for your attention, and please do reach out to Sania if you have more NetREM related questions, because she is awesome and her tool is pretty cool.
Yay.
Any questions? Any suggestions?
My question's just more further down the pipeline already after running the whole thing. Like, what do
Okay.
you use to actually plot the GRNs? This SCENIC offer. I haven't used the tool.
Oh no, I'm not using any particular tool. I'm just doing it in Python.
Oh, okay.
Yeah, I'm not using any particular package for that.
Okay.
Yeah. Yeah, because like the final data set that we have is kind of like an amalgamation of a bunch of different methods. It was just a matter of whatever tool that help us make the nodes and then the edges and then introduce all of these variables. Because then later we're like, oh, we want to add the GWAS hits. We want to make all of this because the network by itself may not be very telling, but then you want to like incorporate all of these other interactions there. Yeah, so I was not really using like a particular just
So like
graphic.
this plot is just made in Python?
Yeah.
Oh, cool.
Yeah, just graph. Yeah, just Python's basic functions, not a particular package there. So I think it would be nice to have like a more complex visualized tool for it. Yeah. Yeah, this was tough because like my initial plots were really ugly. Like they were just like transcription factor and the genes, and then like slowly we're like building on like adding different I guess information in there.
Is there like cell type information that comes after identifying networks, or is that part of the network?
So the cell type part, so we basically did this analysis in all the cell types separately. So after I had like my data set, I already subsetted it to the cell types. And then every cell type went through pySCENIC and then went through NetREm. And then we got the results separately for that.
Can you go back to like the pipeline slide with the ranking and consensus boxes?
Yeah.
Yeah. I'm curious, for the consensus, was that the total union after the top 5 ranking, or was that of whatever the union was, you took the top 300 at that step too?
Wait, can you repeat that?
So the step before you had top 5K.
Yeah.
Right? And then you took like the agreement.
Okay.
And then was that agreement size N300, or was it bigger than that, and then you just took the top 300 to make it more manageable?
No, it was so that's a really good question. But the TFTG links, because you can imagine like every transcription factor is affecting the target gene. So you have like a huge list actually from both methods for the TFTGs. But I mean, these numbers are very arbitrary, to be honest. The 5K was to restrain it in each method to kind of say, okay, like this method thinks these are the top ones, and this one this thinks these are the top ones. And then I kind of try to look for the common, like what is TFTGs that are showing up in both of them. But still that list was like more than a few thousand. But then not as like definitely less than 5,000. I don't remember if it was like 3,000, but definitely more than 300. I would just pick 300 because it just seemed like more probably like higher confidence than other like down the list, basically. Yeah. Yeah, all of these numbers are very, I don't know, like you can decide how to go about it, I guess. But also there were some like TFs that we wanted to capture. So that required me to go a little bit down the list because like there are things that are known that they should show up and they wouldn't show up necessarily in the higher, like when you rank them, some transcript factor wouldn't show up and you have to like kind of go down the list. So when the biology made sense, it's like, okay, we keep it at these numbers, but yeah. Yeah, this is I personally thought including the priors are helpful because when I first ran SCENIC, I was pretty happy with the results and I was like, oh, we can find like activity of all of these genes and how they're, you know, like you can see cool things on the UMAP. But then like learning that, you know, we can use like NetREm to predict these other things. I was like, oh my God, how do you know what you get makes sense? So at least now I'm a little bit more critical of how I should be running these tools and like being aware of the assumptions, I guess.
Or in the like references used for SCENIC that can impact your results in the selection of databases and stuff.
That's true too. That's very true. Like what kind of, yeah, like what kind of transcription factors we are using or for what organ or tissue. That's definitely true because I just used pySCENIC's suggested ranking of the motives and all of the transcription factors that they suggested. So that's definitely that definitely can affect. Yeah, it's just you know, because like they require specific formats too. So like you don't want like I didn't want to like deal too much with like conversion of the file to the other files and I was like okay, but I think NetREm has a little bit more flexibility in that because I think like it doesn't require you to have like a lot of specific formats. Like I didn't know what a feather format is like for running these things on pySCENIC, um but yeah, definitely choosing what database you are getting your transcription factors from is going to affect your results.
Yeah.
Yeah.
I didn't know, I used SCENIC and AUCell as part of that. And then at some point I went into a workshop where people were using AUCell more generally. It's actually a very useful tool for like quantifying collective enrichment of a set of genes, which I didn't really realize. Like if you're someone comes to you, you know, like I want to see these 10 genes, like what is, I think these are important markers. It's really useful.
Oh yeah, for sure. I was a little I guess like I have like this small thing here. Yeah, I mean I think the numbers are pretty small. So so yeah, I guess like you said, like we can like consider any like it doesn't have to be these I guess like data there. We can use like this method to show I guess the enrichment for any other data set. But yeah, I guess like the y-axis are the genes in the regulon. So the regulon is the transcription factor and the target genes that it's affecting. So they only are showing like, I mean, they're showing 4 and 6 just for demonstration purposes, but I guess it's just a funny way of showing how like how confident they are that the regulons that they find are real, because before finding the genes in the regulon, they kind of rank the genes based on their gene expression. So on the bottom, you have like 20,000 genes, but they're ranked based on like how highly they're expressed. So the highly expressed genes are more like in the beginning part. So whenever you find a gene that is in the regulon, so a target gene, you find it, you kind of jump a step. So this kind of shows like, I guess only like there was like one gene that was overlapping with the highly ranked based on the gene expression pattern for this transcription factor, which is why they call it off. But then here, like, yeah, you can see like there's more genes that are found in the regulon that were ranked as highly expressed. So they call that kind of on. But yeah, I guess like it's a matter of finding like, oh, what hits am I finding in this set of genes that I think are important because they're being highly expressed. It could be anything. It doesn't have to be the regulon. Yeah, that's an interesting method that they're using. It took me a while to wrap my head around the scoring. Yeah, if anyone wants to use pySCENIC, I hope they find this a little bit, I guess, helpful in understanding the notebooks. They have really nice notebooks, so once you get the steps down, I think you can follow it pretty easily. Oh,
From Z.
oh, Z.
Mhm.
Oh, yes. So the idea of using the protein-protein interaction is to constrain the list of TFs. So we don't want to pick up just any transcription factor. We want to pick up transcription factors that are already embedded in a network with other transcription factors. So we kind of give it that network. Does that? I don't think she was. I'm like, okay, yeah. Yeah.
Indeed.
Yeah, we can approach. Thanks,
Thank you.
guys. Thank you.